Okay. I'd also like to thank I'd like to begin by thanking the organizers. So thank you for having me. My name is Siddharth. I'm a postdoc with Venki. I'm going to tell you about our efforts to understand how mice track odor trails. And this is out as a preprint, not very long ago, I believe. So if you have thoughts, comments, feedback of any kind, get in touch with either me or Venki. I'm happy to hear them. So the general idea is, and I won't belabor the point too much, the general idea is if you now imagine this road, hopefully what you can see is that it's winding and you have full access to seeing the full scene. So you can see the end of the path and you know how to navigate this on some plane. Now imagine the same thing in a dark scenario where you have to rely mostly on local information and you can't see till the end of the road. So the idea is that you have to rely on local movements and then and locally accessible information and then navigate this. This is the same framework that we think of in odor trail tracking, in that they can't sense far ahead. And because they can't do that, it's not immediately clear where they would, how to go about going forward in predicting this. And that question of where to go becomes very fundamental in this setting. So animals across species, as we've been hearing, do this. This is a moth that is exquisitely tuned to the odor of the flower in this case. And there is also a lot of work about this across species. Since ants have been the predominating theme of this meeting so far, how would a talk be complete without video of an ant? So this is work from the lab a while ago, wherein the ant is essentially tracking the trail. But this starts setting up the question. You can already see the ants comparing to antenna. Comparing the information hopefully across to antenna and making these crisscross movements across the trail. Now mammals also do this. All I want you to sort of really pay attention to is sort of these crisscross paths over the odor trail. It's tracking a pheasant in this case. So the fundamental question is, how do animals rely on local information and follow these surface-borne trails? And hopefully by the end of the talk, you'll hear hopefully you'll have some takeaways from this. We thought we could study this in mice, and here's a simple experiment wherein the mouse is following an odor trail just printed on paper, just presented on paper here. And you can see that it just is not following, going straight. It's making these, it's sensing the trail and making these local corrective movements, and again, making these crisscross movements. So that seems to sort of be consistent across all organisms that seem to do this. And these corrective movements essentially force the animal to then take take a snapshot of the information again. And therefore, the idea is that there is a tight sensory motor loop wherein the animal is navigating this trail. And this provides some key entry points that hopefully we will get at some point in the future. Now, when I said a simple experiment, I was kind of kidding. We over-engineered the heck out of this. So here is an example of our rig experimental setup in the lab, wherein we have multiple cameras pointing at the mouse. The floor is essentially a moving paper treadmill that's pulled from one side. And we use inkjet printers to deliver the odor trail. It's essentially, for the aficionados, if anybody's interested, it's just the smell of rose that the odor is tracking in this case. It's slightly appetitive to the mouse, but has hopefully no innate value. The power of the rig essentially comes out because you can change things on the fly, and you can give you can present trails of different kinds to the mouse. Here's an example of a view from the top, from the camera, and hopefully what you can see is that the mouse is tracking the trail well. The position of the snout is just markerlessly obtained using deep lab cut. And the idea is that the exquisite part of this setup becomes hopefully very clear in something like this, that mice can track trails of different geometries, but it also gives us many sort of stretches of trails to look and study this behavior at quantitative detail.
Is the line visible?
Sorry?
The line visible?
To the mouse, no. It's done in far red. This is sort of post hoc. We've sort of darkened the trail. So we believe the mouse can't actually see the trail. There are also some control experiments that we believe that we've done to address this. But yes. Yeah, and also feel free to stop me with questions. I don't have to get through the whole thing. There's a lot of things here. If nobody stops me, I'll be done very, very quickly. And as similar to Yasuga, I tend to run, so feel free to stop me. So, to talk about experience, mice do this very intuitively, but even then, a moving floor is kind of, not normal to us on some plane. So it is to the mouse. Over about, ooh, that's new.
So.
Okay, cool, thank you. So over sessions, essentially the animals become much better. What you can see is that the animal is like now, following the trail with more fidelity on some plane. This can be quantified. If you just measure the distance of the snout to the trail. The snout is just the tip of the nostril. And what you hopefully see is that the variance of the naive mice is all over the place. It is not, sometimes it goes to the edge of the box and things like that. And by in the expert session, and by expert, what I mean is like in about 5 days, you see that mice track the trail very, very well. To motivate them to track the trails, we also present chocolate drops as rewards. And if you were to look at how much of the chocolate drops they get, again, in about 4 or 5 sessions, which is days, we do 1 session a day per mouse, we find that animals get like about 75% of the chocolate drops. Now you can go in and do something very cool, which is essentially we were doing this as a control experiment to sort of make ourselves, to sort of test whether they are actually just following, getting the chocolate drops. But you can keep everything else the same. You can deliver the chocolate drops at the same, frequency. But you can now take the trail out and see whether the mice get the chocolate drops. And what is interesting is that they don't. So this is very sort of reassuring to us that mice do actually use these odor cues to get to the chocolate drop. Okay, so
just correct.
Yeah.
In the previous lab,
Yeah.
what are those peaks in the naive frequency? Is it just the range of motion? The lateral peaks in the naive?
You mean
in the blue curve?
Oh, in the blue curve. So the naive, this peak is, we believe it's close to the wall. So it's essentially going far away from the trail, and there is a wall there. There is a physical wall there. So great question. So if that was not clear, okay. So this is the edge of the box, right? So they can, these are all expert mice, so you don't see them, but like they could, in principle, hang around next to the walls themselves. Yes, sir.
Can you distinguish, this is maybe a dumb question, can you distinguish between learning to track the trail versus learning there's nothing else? Like they stop looking off the trail because they...
Yeah, to some extent we can. So if, let me get to that. And if I don't answer that in a couple of slides, we can come back to that. The general idea here is that, because we have a secondary side camera from the side, you can also quantify how they sample the trail space. And if you look at this, the idea is that a naive mice essentially spends time not just, away from the trail, but also by sampling in the air a little more than actually the trail itself. By the time they become expert mice, they essentially just follow the trail by keeping the snout close to the ground. If you now look at the distribution of the snout in space, you see that in a naive mice, this is very variable, and it's all over the place. And in an expert mice, it's like tightly centered around the trail, but also close to the ground. So on some plane, I believe that if there is nothing else, they would start, doing rearing and other behaviors.
So just a clarification then, is this looks like when their snout is low, even when they're naive, they're pretty good at it. So is this more about learning what they're trying to do as opposed to learning a motor behavior? In other words, do you think there's a lot of like motor learning, or they're already kind of good at that and they're just trying to figure out what's going on?
Yeah, I think there's definitely a component of motor learning here, especially with the moving floor, and that's part of the thing. I believe that if we had like infinite space and we get mice to do this, they would actually follow the trail pretty well. Like the only thing they have to know here is that there is a moving floor. The back of the arena, is a limited space, so they learn to keep their tail up. So there is some level of motor learning here for sure. But
I see. But if you just train them on a treadmill first,
yes,
and then gave them this task, would they just immediately be good at it then, if they were already good at running on a treadmill?
To a large extent, I would believe that that's the case. Yeah. Yes, they are mostly, like, especially the fidelity of the learning is fundamentally them learning things about the arena itself.
I'm not so sure. I think Noah's question is like, if they're running on the treadmill, they have no idea there's a trail.
In the
why-
beginning,
Why
yes.
would they immediately know when you put the trail on? I think his question is
Oh.
about the nut snout, right?
Okay.
The snout,
When their snout is low, they seem to be similarly good at tracking.
yeah. I think the answer is that it is true that they can do it. Right?
Yes.
They're just-
Yes, exactly.
Anyway.
Yeah.
I think maybe we should discuss this. I think there's something interesting here that we should really think about.
Yeah. So one thing you can go and do now, talking about snouts, is because they have two nostrils, you can block one nostril. And if what happens if you block one nostril? This has been done before. The thing here is an example stretch. In black is the trail, and in purple are the dots reconstructed from the snout of the mouse. And if you block one nostril, we find that the animal is biased on the side of the nostril, in that it sort of keeps its good nostril closer to the trail, and you can hopefully see some lateral excursions more on the side of the stitched nostril. But the more interesting question that we were interested in was to figure out whether mice form some model of trail statistics based on experience. And to do this, we kind of performed three types of experiments. One is to essentially introduce breaks in the trail. So the idea here is that you sort of have, trails either that are straight or of different tortuosity. Let's take one tortuous example. And what happens now when the trail suddenly stops? What does the mouse do? Will it just continue in a straight line? Or do they do something more interesting? Another type of experiment that we sort of did was to introduce unexpected bends in the trail. When the trail is straight, now you suddenly have unexpected bends, and this can be introduced at different times. And the last one is to essentially do an experiment wherein we keep the trail straight for a fixed amount of time, but then you also introduce different kinds of wiggliness before that. So, here is an example where it's straight, then you have a particular convoluted trail, then it's straight again, and then a different tortuosity, and then it's straight again. And then look at, in these sort of straight or probe sessions, how does the mouse actually navigate this section. So, coming to the first part, here is an example of what happens when there is a break in the straight section. The animal essentially makes these casts that sort of increase. What is interesting is that when you basically have a trail that's more tortuous, the casts open up faster. And if you were to just look at the side-to-side extent of the cast, you find that coming from a tortuous trail, this is opened up faster. This is also a prediction from our friends who wrote a paper about a sector search, which is a strategy that animals could use to essentially follow ground-based trails had. So it was kind of like a nice test of a theory if you were to take that. What about unexpected bends? In this case, here is a section wherein the animal has been tracking the trail for about a second or so, and then there is an unexpected bend, and a longer stretch, and a longer stretch. And what we find here is that fundamentally mice take these excursions from the trail. The post-turn deviations are much larger if they have been tracking the trail for longer. This is not a condition of velocity because we've actually checked the velocity between it, so it's not purely inertial. And there is no difference in respiration as such when the animal essentially makes these. So we think that it's kind of like an expectation that the animal has that, if it's been following the trail for some amount of time, it's confident that the trail is actually going forward, and therefore when the trail unexpectedly bends, it still continues with a larger excursion before coming back. Coming to the last straight probe session, we find that, if you were to now look at sections where the animal now will have a straight section, in the straight section, the lateral deviations seem to be larger if the preceding trail was more convoluted. You can now systematically do this, and we find that, we find a nice relationship between the snout deviation in the straight section being larger based on the preceding trail as RMS.
Not consider the first part of the straight trail, because obviously if they're coming in following something curved, they might just be sort of naturally overshooting.
But for these quantifications, we've essentially taken the entire straight section. And we find that actually, so if you split them into 3/3, we find that it's kind of towards it is mostly the first 1/3 that you do see it, and then it sort of flips down. We've also done experiments where we introduced just one section of convolution and then kept the trail straight, and we find that mice slowly, sort of come back down to like whatever the baseline is. So what I've told you so far is that based on experience, animals get good on following the trail. And also that information from both the nostrils are essentially important, but most importantly that they used a recently encountered trail history to essentially form some kind of policy about following the trail itself. So how is it implemented in the brain? I think that's what I'm interested in fundamentally. So as animals take an inhalation, odor molecules bind to receptors that are expressed in sensory neurons, the OSNs, and these axons terminate in anatomical units called glomeruli, which you can find in the bulb. So one idea is to essentially put a microscope, express some calcium indicator, just like Ben was talking about in his flies, and essentially just image the glomeruli themselves. Before we get to that, as I was talking about two nostrils, we all have two hands, two legs in symmetry on some plane. The idea is that there are projections that take information from one hemisphere to the other, and in the rodent scenario, the olfactory information is kept as two independent streams until it reaches this early cortical area called the anterior olfactory nucleus, where there is this fiber bundle that essentially takes him to the other side. So this is like literally the first site where bilateral integration happens. So what happens if we now keep the streams separate, but cut? So for simplicity, essentially if we sever the anterior commissure, what actually happens? This is an actual image from a horizontal size. That's the AON, just to give you a feel for the anterior commissure itself. And this is the fiber bundle from one slice. So hopefully what you can immediately see is when we cut the commissure, animals are actually very bad. They don't really follow the trail. And compared to sham experiments wherein the animals can still follow the trail just fine. So these are just control experiments, and this is where the commissure is actually cut. And you find that the lateral excursions are really large. They are fairly bad. They don't recover. This keeps happening. Like we've run them for like about 2 weeks, and they still do not recover at all. So they really don't know what's happening. And one idea was that because now there are two nostrils or two streams of input, is cutting the commissure somehow messing with how these representations are matched on some plane? And to test this, we tried to block one nostril in this scenario. So in other words, you take an animal whose commissure is cut, which is two independent nostrils, which are both open, and now we shut one nostril. Very interestingly, we find that this can partially rescue this behavior, suggesting that the sort of thing I was telling you about two different streams getting put together is important. These animals are actually surprisingly fairly good following this trail. I mean, they still have like larger deviations than like sham, for example, but like they can still follow the trail as opposed to just the commissure cut animals. So this was actually very cool. This is of potentially interest to us, but I don't have a lot more mechanistic insights into this at this point. This is just to quantify the peaks, and hopefully what you can see is in the AC cut animals, there are these peaks, and the minute you shut one nostril, it looks pretty much like sounds. Okay, so now imaging glomeruli. So what do the sensory responses look like? So this is a video acquired by a microscope that's sitting on the head of the animal. We express calcium indicators, which we use as a proxy for neural activity. And here is the mouse tracking the trail. Hopefully what you can see is like, some of these glomeruli flashing when the animal is following the trail. If you look at this, on top are just neural activity from glomeruli. Each red vertical line is an inhalation, and the green lines are inhalations that somehow seem special because they are the ones that seem to sort of elicit a response in the glomerulus. Obviously, if you now look at behavior aligned to all of this, we find that there are some of these inhalations close to the trail, which evoke these responses in this single glomerulus, for example, which is an example glomerulus in this case. The only thing I want to say here is that the response probability of glomeruli essentially are higher in animals, especially expert animals, close to the trail. So in other words, the expert now has, there are sensory responses close to the trail when the animal is actually tracking the trail well. And because I was talking about the anterior olfactory nucleus, the idea was to essentially do acute chemogenetic inhibition experiments. And if you were to inhibit the AON, you find that the animal tracks the trail poorly compared to the same animal when you don't provide the ligand. So again, the point here is that we express a specific type of ligand-based receptor in some of these neurons bilaterally in the AON, and we shut them down by providing the ligand, so you can inhibit those neurons acutely. And if you don't, you find that the animals are fine. So in this case, it's the same animal as opposed to the sham, the AC cut, which are different sets of animals. So now what happens if you were to put a microscope in and look at neural activity in the anterior olfactory nucleus? Here is a population field of the neurons, and the flashes that you see are, the neurons firing on some plane. And the idea here is that if you now remember both the turn experiment as well as the convolution that we provided and looked at the straight sections, we find that here, post-turn response similarly seems to correlate to how long they have been tracking the trail. So in other words, if the animal has been tracking the trail for longer, the post-turn response actually is higher, which hopefully you would remember from the behavior. If you don't, it'll come up. And similarly, the response in the AON seems to correlate. These are in the straight sections to immediately what they have faced in the recent past. So if the animal comes to the straight section and the trails are preceded by a larger RMS, the responses in the AON are higher than if it was like relatively straight or smooth. So this is actually very interesting because when I went into this, the idea was that we probably see only sensory kind of responses in the AON. And this indicates that it's not entirely sensory. There's definitely something more than sensory, maybe sensory motor. I don't know at this point. But we are excited by this because going forward, I think the idea of where in the brain these memory, by memory I mean some context-dependent information about recent trail experience is implemented is a potentially interest to me. And this is just to give you, to sort of bring back the idea that here is the imaging response, here is the behavior from a different set of animals, but you can see that it sort of correlates well with behavior. So that's kind of what I have so far. That's where the story is at this point. And the idea is that we believe that this trail-dependent, the recent trail experience to future behavior is put somewhere together, and potentially the AON is the start of an interesting place to look at. And with that, I'd like to thank people involved, Venki, the other people in the lab, theory collaborators, especially Massimo and Nicola, who were involved in this project, the neuroengineering core, and funding sources.